I think it's really important in the early days to validate what you've got with real-world applications and real-world samples. And to me, we did a lot of kind of research services and a lot of basic research early on, but the biggest success out of all of that was validating real applications in oncology, to prove out not just the technology, which is important, but to test out and prove a market.
Hello, everyone, and welcome to the Health Technology Podcast. My name is Christine Winato, and I'm the director of the UCSF Roseman Institute in San Francisco. Today, we're talking to the CEO of Mission Bio, Charlie Silver. Charlie comes to healthcare from an unusual background. He was an astrophysicist, and earlier in his career, he worked on instruments to look deep into the universe and observe the cosmic microwave background. Now he's looking inward, focused on much smaller things, single cells. His company, Mission Bio, recently raised a $30 million B round to help launch its single-cell DNA sequencing technology. Mission Bio's product uses microfluidics to tag cells with DNA barcode. This enables researchers to get a handle on how diverse a population of cells is. It is the variation in the cell's DNA that sometimes enable cancer cell in a tumor to escape chemotherapy. As you probably know, there's a lot of diversity in how companies get funding. Mission Bio is no exception, and you'll be interested to hear how Charlie fueled progress early on by a mix of grants and contract work before talking to investors. I hope you enjoyed the talk and you like what you hear today. Please subscribe to our podcast on iTunes. Welcome, Charlie. Thanks for joining me today. It's good seeing you after, it's been a while since the last time I saw you in the hallway.
Yeah, it's great to be back chatting with you.
Yeah, so, you recently raised $30 million for Mission Bio, and I think that's really, when I saw that, I was like, yay, Charlie. I always thought it would be great to start with a bit about your background. Many of us, I know you, but, it would be good for the listener to know how you end up in Mission Bio and what was your background? How did you get where you are today?
Yeah, I have, a background in a very different space. I studied astrophysics in college. And I actually double majored in astrophysics and philosophy because I was very interested in how things work. And I also studied astrophysics in grad school. And what always excited me about that field was the ability to leverage what we know right here and ground truth about the world around us to project what we can learn from the world way, way out there. And this idea of kind of using the very latest and greatest hardware and technology that we can build, the very latest technology that science has to offer us, to build hardware that can see further and deeper than anything anyone's ever seen before, has always excited me. So in astrophysics, I was always on the experimental side, building hardware. And that's very much what I've done throughout my career. I've always been excited about that next layer of resolution or the next level of seeing that gets you deeper insights.
So you were not in the healthcare space before Mission Bio?
Yeah, started in physics and really hardcore astrophysics when I was in college.
What is that? What kind of work that you do as an astrophysicist?
So, look, what astrophysics does is study the stars and, the depths of the universe to understand how the world started. Ultimately, it's about looking as far out as you can to the edge of time and figure out how we got here. What I worked on was really the hardware that enables that. So there's a theoretical side, which is math and equations that I was never very good at. And there's a hardware side, which develops the instruments that give the tools of being able to see deeper. In college, I worked in a lab of a next generation gamma ray telescope. Where we built a telescope that went up on a balloon flight, and I was part of that. And ultimately, it was the hardware that went up on the next generation satellite that's in space. that was used for collecting gamma rays from distant galaxies and understanding better physics about those. And then when I was in grad school, I was part of a bigger group that built detectors that looked at cosmic microwave background, which is a way of seeing to the far edges of the universe, which is within the first few microseconds of time after the Big Bang. So these are very sensitive detectors for seeing way out. And I was really privileged to be part of the team that built the hardware for those, that also went up on a balloon flight and ultimately and ended up making measurements that told us really important things about the edges of the universe.
That's really cool. That's really technical and seems really way out there.
Yeah, I've always been excited. Well, I mean, it's technical from a hardware perspective. I've always been very excited about building the hardware that lets you kind of see that next layer of resolution.
Yeah, so it's so how did you end up joining Mission Bio and lead Mission Bio?
So I was kind of a story about where I came from. I went to graduate school in astrophysics. I was the youngest of 6 kids. The 5 older ones were all doctors and lawyers. And when
the dreamer.
When it got down to the youngest, my dad was a PhD. So when I started in a PhD program, that was his dream come true, because all the rest of them, he'd done that. Now he had the PhD to follow in his footsteps. And I decided that academic life was not for me. I'm good at building hardware. I'm good at building at problem solving, but I'm just not satisfied by writing papers. So I left grad school. And when I left, I made the commitment that I would use that sort of energy to build things, to really build the tools that enable scientists and engineers to make the discoveries that they're very good at. And that's what I've spent a lot of my career doing. My last company, I've been at a handful of startup companies. My last one was an electron microscope, which was these are workhorse instruments of the engineering and science industry. But we built a new type of electron microscope that kind of democratized electron microscopy. So anybody in any lab could use them. We actually placed one of the first ones, I think, actually here in Byers Hall at UCSF, up on the 3rd floor. So I was really privileged to be a part of that team.
So were you starting that company? Is that your first stint with the startup, or
I was there behind the 2 founders. I was the first employee at that company, and I ran engineering and also product management.
Okay. And so you were doing that and hang out at UCSF a little bit because of the products being placed here at UCSF.
I did. I spent a little time here and at other campuses because those were the early customers for us. And ultimately, that company was picked up by Agilent.
Okay.
So I spent a couple of years at Agilent and then really looked around for a technology that leverages, what I've done before, which is bring together diverse sets of people, but especially complicated systems with all of the latest cutting edge science built into them, to be able to see deeper and get deeper insights. And I really wanted to apply that to the healthcare space. Recognizing that I think there are a lot of tools that physics and engineering give us that I think haven't really materialized as well into what can be delivered in the healthcare space. So that was really my mindset, and I ended up connecting with Adam Abate in the bioengineering.
So how did you get connected to Adam?
It was through the network. Actually, Doug here was the one who originally made the connection.
Okay.
Doug Crawford. But again, it was a connection through the network. I was looking out for technologies that kind of align with what my background was, that really have the potential to make just game-changing differences to the healthcare space.
Yeah, so I'm trying to remember, because earlier we talked about this Mission Bio. The idea start a while back ago. And when did you join Mission Bio?
So I joined up with Adam in 2013. And at that time, Adam had started as faculty here in 2011, built out a team into his lab and started this technology incubating in his lab for a couple of years with the intention of spinning out a company behind it. So when I joined up with Adam in 2013, I spent about a year or so working up the business case, figuring out where the market wanted to take the technology as they were on the bench, getting it to the point that we could start running outside samples, which is the real validation for life science.
Yeah.
And when those two came together, we got the company spun out in 2014.
Yeah, so maybe this is a good segue to tell us a bit more about the technology. Because at least, can you describe to us what is Mission Bio working on and why this technology matters?
Yeah, so at a high level, what we're trying to do in biology with genomics is understand the drivers of disease. And for complex diseases, like cancer is the typical example that we use, we know they're driven by genetics, but it's not the DNA itself that drives it. The DNA is a molecule. It's the DNA working inside the cell that drives the disease, and the cell function is what actually causes the disease to progress. But it's differences among cells that are causing the disease. In other words, this cell has some DNA change that's different from this cell, that's different from this cell. And those changes and the differences among all the cells make it very hard to treat, because you hit the cells with one drug, and some cells remain because the drug is targeting one mutation and the others come back stronger. And that's really been the challenge with treating cancer as long as we've been doing this. And the challenge ultimately is being able to look at the DNA in every cell and understand what's in there. So that's sort of the problem statement behind what we've done at Mission Bio. And it's a problem for all of biology research. Ultimately, you're trying to understand, cell function based on its genetics, which is a single cell phenomenon. So what we do at Mission Bio is single cell sequencing. And the technology that enables it is droplet microfluidics. And that's what Adam Abate, has built his career around. He's one of the top leaders around in droplet microfluidics. And this is basically a way of making the smallest test tube you can possibly make around a cell. So these are, think cell is microns in size. This is a few microns, right? The very smallest test tube you can put a cell in, but then make many of them. And by many, I mean thousands, millions of test tubes. Every test tube gets its own cell. So instead of looking at the sample in aggregate and an average across all of the cells, you're running reactions independently on every one of those one at a time. But because the test tubes are so small, you can run them all in parallel at a very low cost because the reagent volume is small enough inside those test tubes. So these test tubes are droplets, and it's an emulsion, sort of like your salad oil. And it's literally the same phenomenon. And the droplets are made in an oil carrier in microfluidics. So these are channels on a device that, think they're microns in size, makes a micron-sized drop that encapsulates every single cell one at a time inside these drops, but does it fast enough that, over minutes or an hour, you can process thousands, millions of cells at a time.
Okay. And then once you process that cell, what does your device do? So
the technology we built is a way of using those test tubes to put a unique barcode sequence into every cell and enzymatically amplify that barcode sequence up against the DNA of the cell. So that barcode, which is unique to every drop gets its own barcode. It uniquely labels the cell. The barcode is a stretch of DNA. So we synthetically manufacture these unique sequences of DNA. Everyone gets a different DNA section that then integrates with the DNA of the cell itself in the drop. And then at that point, we can take all the DNA out of the drops and throw it on a sequencer. And the sequencer doesn't know what it's doing, right? It just thinks it's sequencing through DNA, but it actually sequences the barcode and then the DNA of the cell. And then informatically in the software, we can reconstruct which read came from which cell, which is a way of giving you single cell sequencing.
Okay.
So the technology, the product is an instrument that runs this technology. The instrument runs these microfluidic cartridges that have those channels making the droplets in them, together with all the reagents and enzymes that it takes to run the reactions. And what comes off that is what then goes onto the sequencer and then runs through the software that we provide at the back
And
end.
I assume you can use off-the-shelf sequencer.
Yeah, any Illumina sequencer. We built off all the Illumina sequencers, which is most of the sequencing deploy base for our customers.
And then once you get the result, the analysis, then you can share that result with the end user customer with that particular report.
So we built this around an instrument consumable software. It's the razor blade model, where we send the instrument out. That's the product that the customer buys, and then they buy the consumables that run on the product, and we provide the software to them.
Okay.
So they're running this on their bench, and they have the software that we give them to be able to analyze it. And it's an all-in solution, kind of fully contained.
Okay.
That we provide them.
And who is them? Who is the people who bought your?
Our customers are very much on the translational side, and we entered this single cell market with the mindset that there's a strong research side of the market that's already being very well served, and that's becoming more and more mainstream. So single cell sequencing is something that the space has been looking for a long time, and it solves a big problem on the research side of the space. And we recognize that research is now going to have need to translate into the clinic. And when you think about the clinic, you think about actionability, number one, and of course, workflow and cost. And those three dimensions are really important in a translational product that will then move into the clinic. So in terms of actionability, what really matters is DNA. Because that tends to be the analyte that can move into the clinic, because it's robust, it's easy to make an analytical and quantifiable measurement off it. So DNA is important, and that's where we differentiated from the rest of the space. And then we have a workflow and cost that really keeps the costs down. So with all of that, our customers are at cancer centers and also drug companies.
In the cancer center you mentioned, so it's like a research tool for these people, or they're really using it as a clinical, help them to make a clinical decision?
It's an RUO product, so it's not being used for clinical decision making, although it can be used to support decisions as part of a larger data package. It's been on the market since the end of 2017, so we've been selling for about 18 months at this point. And it's used really for translational research studies. And by that, I mean patient samples that are collected by the MD PhDs at cancer centers, very often funded by pharma studies. So these can be, pharma funding that are running early stage clinical trials at the cancer centers. To look for biomarkers that identify mechanism of action, mechanism of therapeutic resistance among patients in a cohort.
Do you envision your product eventually will be used more to make an impact on a clinical decision?
We do think so, and we're really orienting the business in that direction. Where the market is at right now, there's a lot of activity in the translational research side of the space on our platform, running these studies that prove out where it will land in the clinic. But we do expect that this will be used in the clinic.
This podcast is sponsored by IO Life Science, a Silicon Valley-based medtech design and development consultancy, transforming your technologies into a unique healthcare solution. For more information, visit their website at iolifescience.com. This podcast is also sponsored by Brown Ratnick's Global Life Sciences Group, a team of legal professionals that helps life science companies, lenders, and investors around the world turn good science into good business. Learn more at brownratnick.com. Yeah, so now going back to different topics, you raised that $30 million. I'm sure there's a lot of ups and downs when you raise that $30 million. And people always say the first money is always hard, but people, at the same time, I always think the next money is always as hard too. But let me know if it's not that case.
I think every round has different challenges, really because there are different milestones. Early on, you're proving out the technology and building a team. Later on, you're building out the market and really identifying which market is going to scale up in a big way. So I think every financing is really just different because of the stage of the company.
Yeah. So when you first joined Mission Bio, the first round you had was around in the $3 million. Like who did you reach out to, and how do you manage to get that funding?
Yeah, that's right. We raised an early seed round that we actually started even when we were here at UCSF, but that was even behind a pretty substantial amount of government funding. So the founders early on got very good at writing SBIR grants and pulling in government money for the core technology development. And that was sort of one vector of funding the company early on. The other one was running services. And early on, I mentioned we were spun out as we were running real world samples, and we ran that as contracts for pharma partners, where they would run, pilot studies, co-development, validation studies, send us the samples, we process it on the breadboard platform, and then send the data back to them.
It's almost like what your customer is currently doing.
Yeah, very much. Before we built the product, we were running this as a service. So all of those early sources of funding were non-dilutive and really got us, pretty far along before we had to start raising the seed money. And, for a life science company, very typically you'll raise the Series A to start building the product, the Series B to launch it. But all that early source of non-dilutive funding meant we were able to really shake out the technology with real world samples before we even raised money. So we kind of jumped ahead of that funding cycle.
And looking back, is this something that you do because you think it makes sense, or out of necessity that you have to do that to keep the company going?
I think it's a little of both. I think as a platform technology that has a lot of different applications in a lot of important markets, and you need to identify which is the one that's going to scale in the way that you want to build the business, I think that early funding is actually really useful to shake out different applications. As we watched the market develop to figure out where we wanted to really lean in as a product. And I mean, we tested everything that you could possibly imagine with this foundational technology of high throughput single cell PCR. An early version of the technology was built to be able to basically flow sort cells off PCR markers. So it's a way of doing FACS off genomic markers. And we ran services off that product. We called it PACS, or PCR activated cell sorting, to mirror FACS. We built a qPCR version of it. We partnered up on circulating tumor cells. Actually, we had a large contract with a pharma company doing circulating tumor cell detection with the early version of the technology. And we played with all different analytes from DNA to RNA to proteins. And we had that kind of flexibility to move in all directions as a research stage company before we really had to zero in on the one that the market wanted. And I think that really gave us an edge up as we got further down because we knew exactly what to build.
It's almost like you're being paid to test the market.
Yeah, I think that's right. And, look, that's the way NIH funding, that's the way SBIR funding is set up to seed early stage technology. And I think we really benefited from that.
Yeah. So, and then you got that early stage seed funding. And then how do you get your first institutional, like the real big ventures? I mean, I know Agilent Ventures is in your fund now. Kodak Capital, that's how Subra Jain, who was on my guest.
Subra, of course, yeah.
Has suggested me to invite
LabCorp,
you.
Yeah, LabCorp and Mayfield as well.
Right.
So early on, behind the grants, we raised some seed capital, which was a collection of angel investors and angel groups from all over that really funded the early productization. That got us through alphas, through an instrument that was pretty close to the version we finally launched. And that was the proof point that investors needed, together with the early services to validate the market, to show that we as a team were able to build the product that could serve this market. So all of that were the proof points that led into the Series A, and that one was led by Mayfield.
Okay. And so now you raise your big money. what is that milestones that you have to achieve?
So this is all about scale up for us at this point. We've been on the market for, since last year. It was our first full year. And it's all about, building the market into cancer centers and especially into drug companies to help them accelerate their drug pipelines. It's all about accelerating product launches. So we built a really high performing team, and I always argue one of the top teams around to be able to launch this product from an R&D perspective, but also a really top commercial team to sell. So it's all about scaling up the product.
That means you do a lot of hiring for your sales team.
We do. We've done a lot of hiring over the last couple of years, for sure.
Yay, more job. So I want to touch down a little bit on the lesson learned throughout this. You've been working on Mission Bio for a while. What are the things that you learn from this experience, and also challenges that you face that you never thought you would be able to overcome, yet you did? And how do you do that?
I think a couple of lessons learned from early on are, number one, as a company in the life science space, competing with a lot of noise in the life science space. There are a lot of technologies out there to do a lot of things. And
everybody talk about single cell.
What's the right thing for your application? Because they're all right for different applications. And a lot of noise about new technologies coming on the scenes. The life science industry is hit with new technologies from every side all the time. I think it's really important in the early days to validate what you've got with real world applications and real world samples. And to me, we did a lot of kind of research services and a lot of basic research early on, but the biggest success out of all of that was validating real applications in oncology, to prove out not just the technology, which is important, but to test out and prove a market. So I think that was one really important lesson, even as we were spinning out of UCSF.
Do you think that helped being at UCSF? We have one of the biggest cancer center.
It did, for sure. I mean, having access to this ecosystem, to the talent, and to the partners to help us validate the technology really did make a difference. And I know, we together with the Abate Lab have leveraged that in a big way. I think, UCSF, and look, I mean, we work with a lot of research institutes all over as our customers now. UCSF really does have a unique position as sort of a synergy of the entrepreneurship community side by side with the real world medical community, sort of feeding each other in this healthy feedback loop. And I think building this technology as quickly as we did, I think we really did leverage that resourcing out of UCSF. And, part of it is just that, the entrepreneurial mindset coupled with the medical community. But part of it really is that sort of entrepreneurial program that you guys have built out here, which I think really does feed a very active ecosystem. So we've really benefited that.
That's great. So, what are the things that when you first joined Mission Bio, looking for funding for a lot of early stage entrepreneurs is always the one that is very challenging. And what advice would you give somebody who just started looking for their seed money?
The best advice I could give anyone is to hire the very best people as early as possible. And I say that from experience. We have a very complicated product that leverages many disciplines, all packaged up in a very, very compact form factor. And when we hired in really top leadership across each discipline, I mean, it did a number of things. It got the product off the ground in a big way. It enabled the founders to really, move into a mode that they were moving the product forward much faster. It just moves the company much faster.
And how do you get the best people when, funding is tight? And, how do you compete with the bigger company who can afford to pay the big dollar?
I think it's the vision of what we do. We really are about enabling our customers to cure cancer. And our customers, we hear this all the time, our customers very explicitly buy the product because they believe our products will enable them to cure cancer. And we bring those stories back into the company. They come back and talk to the whole company. And we talk about that all the time, very openly. And that's part of the culture of being at Mission Bio. And it's part of our mission and values is to support our customer on what is a really important journey. And I think as a startup company, I mean, look, you're competing for talent from all over. And we've been able to hire just the very best scientists, engineers, leadership from all around, and all across the company, manufacturing, operations, G&A, all the very best people who could be working anywhere else, but they really believe in working together as a team on the mission to help our customers cure cancer.
So, as I'm sure that your company has grown, and how do you maintain, or I'm sure there's every company has their own culture. How do you keep that culture stay intact as the company grow?
It's hard for a company to do that as we grow, but it's important to be very mindful about it, very thoughtful and deliberate before the company grows, because after it's grown, it's too late. And ultimately, a culture is literally how you define it. Because it's very mechanistic. It's a way of hiring people. These are the people who match your culture, and ultimately, the only way you have to make sure the culture stays in the company is to apply that across all of your hires uniformly. And what that means is, as we're interviewing against our culture, we're making sure people match what it means to be at Mission Bio. And that means everybody at the company shares the same values. We are a very diverse team, and that's baked into our culture. And it's baked in thoughtfully because it's a product that brings together a very diverse skill set. It's a very complicated, heavily engineered product that simplifies the customer's workflow by building complication into the core technology. And it does that by integrating the very best biochemistry, cutting-edge microfluidics, cutting-edge instrument engineering, informatics, software, you name it, it's packaged up into the product. And it's important to bring together a very diverse team around that because the whole team is uniquely very good at what they do, and that enables the team to build something that's much better than all of the individual parts. So we're very diverse. And as a culture, we're very driven by the mission of helping our customers make very important discoveries and accelerate cures for cancer. And we're also curious, and that's an important mindset because our customers are the ones who are making the very latest and greatest discoveries, and they're publishing some really important work on it in top-tier journals. And it's important for us to stay curious about what our customers are doing so we can serve them the best way possible. Because, for us, the customers really are the heroes. They're the ones who are using this awesome technology to make their discoveries.
You're the enabler, in a way, to make that happen. And sometimes it also can be a challenge when you're a bit more removed from the patient. How do you motivate the team so that they know that they're doing something that's making a huge impact, right? And I don't know if that makes any sense.
It's interesting. When we were spinning this out of UCSF, the founders came together and realized, okay, like we're typically, on a bench in a grad student or postdoc lab, you're 5 or 6 steps removed from a patient. You're doing some basic biology that might have some implications down the line, and there's always some, statement up front about the impact, but it's typically pretty well removed from patients. And we made the thoughtful decision that in this market, there's a lot of activity on the research side, and that's really important. But for us as a company, as a really mission-oriented company, we want to make decisions that'll really improve patient care. And we want to enable our customers to be able to do that. Whether it's a drug company that's pushing drugs through the pipeline faster to get medicines that are needed for patients, or a cancer center that's running a study that derives better insights from patient samples. Everything we do as a company is really one step removed from patients. Our customers are the ones who are treating patients every day, and they're using our technologies to study their patients.
Last question. What would you tell yourself now that you have gone through all, the successes and the challenges when, the Charlie Silver who just started Mission Bio? One thing that you would tell yourself.
One of the things early stage companies tend to do is be very cautious about how quickly they scale. And I think there's a really important balance around being cautious, testing markets, testing technologies, developing it to the point that it provides meaningful value to a market that needs it. And then when that opportunity comes along, let it go. Build it as fast and as big as you can because that market needs it. And I think that balance is hard to recognize early on. I think it's a lot easier to see in hindsight. But I think, thinking forward about how this market develops, putting myself in the shoes, in my shoes many years ago, I think I would have probably had that mindset early on around, you need to do the research, you've got to get it to the point where you can deploy it in the market. But when you see that market, raise the money and go in fast. And I think that's a lesson that a lot of entrepreneurs probably learn the hard way, that when you see the opportunity, don't be shy about it. Jump in as fast as you can.
Yeah, well, that's great. I promise this is my last question. Books that you read that you can recommend?
Oh my gosh, books that I've read. I used to, sorry to say this, I used to read 1 or 2 books a week many years ago. And I barely watch TV anymore, and I barely read books anymore. The best book I've read in recent years is, my son is now 8. A couple of years ago, he was 6. And we read cover to cover all 7 volumes of the Lord of the Rings trilogy. There are actually 3 plus The Hobbit. And I'm sorry to say it's not related to the domain,
No.
but these are some of the best books out there.
That's great.
And I'd say reading it with a 6-year-old sort of brings out every dimension of it because it's a great story. It's biblical, it's epic. The language is so powerful. But it's a mirror of everything we live today.
Okay, no, that's great. No, that's so thank you so much. It's been fun. Thank you for all your time.
Cool. Thanks a lot.